Hello.
Hi,
Hi. Who's
Trophy
that?
back here.
Yeah, Harikrishna. Hello.
Hi. Oh, yeah, hello.
Hello.
Hi,
So,
Harry.
yeah.
Hi. Oh, boy.
So, welcome to the meeting. And I don't know where the rest of our GSoC students are, but just Harikrishna wanted to give an update as to where he was. That would be good.
Okay. Yeah, so I'll give my update.
Okay.
So this week I couldn't do anything much extra, but in this week, first of all, what I did was I tried to make a README file. I documented the things in the stages which I did till now, and also um, I worked a little on the UI part of the application which I was building because I couldn't do much because my semester got over and I was traveling back to my home. So most of the time went traveling and packing. So
Okay.
yeah. So in this now next I'll be focusing on the projection of the images. That's what I'm doing now.
All right. Well, that's good.
And I'll also update the GSoC repository today in the GitHub.
Okay.
The things which I've done till now.
Yes, let's take a look at that. Let's see. Okay, so this is the GSoC repository, and this is the one here. Is this one?
Yeah.
Okay.
This was the initial update which I gave. I'll update it today also.
Okay, so this is the README, and then weekly update, image processing, Python. Okay, that's good. And then you have some code here. All right, that looks pretty good. Yeah, so that's good. And then yeah, okay. Let me stop sharing. Well, that looks good. So your plans are to do some work on image projection next week and start getting that done.
Yeah.
Okay.
I also discussed it with Karan, the methods we could use. So yeah, we are working on it right now.
Okay. I guess trial and error, just working on it like that is good. Seeing what you can get to. Yeah. Okay. I don't know. Yeah. So that's all. I think that's the only update we have this week. So yeah, I have, and I don't know if it's for this project or not, but we can extend the projects. Actually, I think it's for all projects. We can extend the projects into the fall if people need to. So if you're needing to have an extension, I just need to know so I can let them know. I think it's just a matter of being flexible on the part of the program. So they can extend it, I think, as far as November. And then, you just take as much time as you're allotted. You just estimate how much time you need, and then we can do that. So if people are running short on time, or, I know that like in the summer, it gets choppy and you don't get everything you want done. If it's just a matter of you not getting everything you want done, though, don't ask for an extension, just if you don't think you can finish by the August deadline. I was actually a bit confused about that. I thought that only some of the projects ran long, but I think all of them can run 22 weeks if need be. So, okay. So I've got a couple things today. I have some. Maybe I'll show my screen again a little bit here. All right. So first thing I was going to talk about is I found this interesting resource. It's a neuromechanical model of Drosophila. So this is NeuroMechFly, and I think it's run out of EPFL in Europe. And so this is a project where they take Drosophila, which is of course another model organism, and they're using evolutionary algorithms and other types of computational models to model the neuromechanics of the fruit fly. So neuromechanics are when, there's some motor activity and it's controlled by the brain, and they look at not just the brain but also the mechanics of the limbs. So there's physics and there's neurobiology involved. There's also some modeling of the loop between the sensory motor loop between incoming sensation and motor control. So this is a computational model of this. They have a fair amount of code here in this repository. They have it. This is actually based on some experiments that they've been doing. So they're collecting their own data at their lab, and then they're actually doing the modeling. So this is kind of a nice resource if people are interested. So this is the virtual fly here, if just so you get an idea of what it looks like. And it looks like a lot of robotics models that people put into virtual environments. And so this is kind of a nice, and they also have pose estimates from the DeepFly 3D database, and there's a repository along with that. So there are a lot of resources here if you're interested in movement. We also have the movement database for OpenWorm for C. elegans. So, we could do cross-species comparisons there if people are interested. So there's a lot of stuff there potentially to work with. Another thing that I have here is this special issue of Royal Society Interface Focus. And I found out about this weekend. This is run by a couple people that I'm familiar with. So this is, let's see if I can find the guest editors here. I don't know. Okay, here we go. So Adam Safran is one of the editors. Michael Levin, Adil Razi. I don't know any of these other people, but I do know Adam and Michael. So that would be something that would be interesting to submit something to from the group. There's taking submissions currently for this, and what this is it's a special issue of Interface Focus, exploring the role of symmetry in complex adaptive systems across scales. So this is emergent dynamics of biophysics to the shaping of underlying adaptations through ontogenetic and phylogenetic processes. So this is basically what we do in the group. We talk about evolution and development, and we talk about some of these other biophysical mechanisms. And so, it's just a matter of finding like a topic and writing an abstract on it. I have some ideas of how this might work, and we have some things in the group that are, probably good for this. But
yeah, I'm definitely interested in working on that. I'm very big fan of both those people that you mentioned.
Yeah. Well, I thought you would be. Yeah. So it might be like a mashup between some of the things we're doing in the developmental AI group and the other in the lab, and then the stuff we're doing with other stuff we're doing in this group. So it might be an interesting way to go.
I think, I could see that. I think it might be a nice springboard to some developmental AI stuff too.
Yeah. So the topics of particular interest include characterizing dynamical systems in terms of symmetry, preserving theoretical frameworks and models. This is gauge fields and equivariance. So they have these like different topics that they focus on for symmetry in physics. And then there's symmetry breaking in development, which is analogous to that, where there's a sort of an introduction of an asymmetry in development. So when there's like there's the division between the anterior and posterior poles in a two-cell embryo that's symmetry breaking, because you get two different parts of the organism that have a different developmental history. There are other symmetry breaking events along like the midline between the left side and the right side of the embryo. And then, this continues on into post-embryonic development. So there are a lot of things that, you can look at there. Symmetry making, and they give an example of morphogenetic fields, which we've talked about as being kind of not existing, but, that's the way they refer to it. And breaking, potential constraints on regeneration and morphogenesis. I think that was something that, that might be something we could address. Asymmetries in cognition. So there's psychological arrows of time and brain organization or hemispheric differences. So the brain itself has a lot of asymmetries in like processing and things like that. Symmetries as inductive biases, identification of invariant features, and objective functions for natural and artificial learning systems. So this has a lot to do with different types of processing and, symmetry and asymmetries. And I think they want to move towards like simulating this stuff. So it's like, they want to identify a lot of these things, maybe computationally. I think that's probably where a lot of the papers are going. The diverse roles of symmetries in cognition and behavior. These are generalized models of inference and coupled systems, mirroring and coordination dynamics, or models or affect across brains and organisms. So this is, again, kind of like this idea of complex systems and active inference, something they call active inference, and looking at that in cognition and behavior. So I think there's a list of papers here, if I can find it. This is the first page I was on.
Bradley, can I suggest something?
Yeah.
Out of Harikrishna's work might come a discovery of what causes the position at which a differentiation wave starts. And that would be a key to symmetry breaking.
Okay.
Okay.
You mean in space and time, or?
Yeah, in space and time. I mean, the wave starts. Each wave starts someplace and ends someplace, and where it starts and where it ends are speculated to be caused mechanically, maybe by stresses and strains bearing over the surface, and that would then cause symmetry breaking in the embryo.
Okay. Yeah.
Okay.
Yeah.
So we have the possibility of measuring strain, and if we resurrect, Wayne Broadland's program for figuring out stresses, we might be able to get that. So, in other words, there could be a mechanics of symmetry breaking.
Yeah.
Okay.
Yeah.
And I suspect, and that would also clear up this the nonsense about morphogenetic fields.
Yeah. Yeah. Yeah.
I mean, maybe it was a good concept 100 years ago, but right now I think it obscures thought.
Yeah. Yeah. Okay. Yeah. So if you go through some of the papers that they have here, what they need is like an abstract at this point, and then they move on to like
what's the deadline on the abstract?
It's open, but it's probably as soon as it looks like they were getting some submissions, but as far as I know, it's still open. So I can find out I don't think there's a hard deadline.
I'm not sure we can get that far.
Yeah. Yeah.
On their time scale.
Yeah.
We'll see.
Yeah.
Okay.
Yeah. So yeah,
maybe if we can get somebody involved in resurrecting Wayne Broadland's work.
Yeah.
I think the program is called CellFit.
What is it called?
CELLFIT.
Oh, okay. Yeah, it's like a program or something.
Yeah, it's a program. He's retired now and not doing any science.
Okay.
So getting a hold of the program and getting it resurrected might take some work.
Yeah.
Okay.
Yeah.
But what it did I had a student back when, 1978. His name was Murray Steen, and he did a paper on simply bubbles between glass plates. And we found that we could do the null case. Looking at the curvature of the bubbles, we could figure out that they had settled down to zero forces between them. Okay?
Yeah.
And Wayne generalized that to an inverse finite element method to give the forces between all cells.
Yeah.
Okay, so, and Harikrishna's data will allow that kind of analysis, but we've got to get the program running again.
Yeah. Yeah.
Okay.
Okay. All right. So some of the featured submissions they have so far here, physics of creation, symmetry breaking, inactive inference, and unfolding state spaces, free energy and inference in living systems. So there's some submissions on what they call the free energy principle and active inference. Then they have an elementary model for the emergence of symmetry concepts by agent collectives. So there's some stuff on emergence and agents in like groups of organisms that behave collectively. Then there's Principia Qualia. Is that?
I want to ask a question on the paper on creation.
Yeah.
Do you know if that includes origin of life?
Let's see. Let's see. Talks about the role of contingent structure in history and explaining the organization of biological systems. It looks like it kind of goes into, doesn't really get into the origins of life. It just looks like it's kind of nibbling around the edge of that, but not necessarily.
Okay.
Yeah.
Okay.
Yeah, so
okay.
Yeah.
Is the paper available?
Oh no, this is just the abstract. This is like, kind of what people are working on. Yeah. So the paper is forthcoming.
Okay.
Yeah.
Do me a favor, send me a URL for the abstract.
Yeah, I will. Okay. So
I'll check further to see if they're looking into that.
Yeah.
Because it might have been part of origin. Meinik, Deb, and I and Natalie are working on this book on origin of life.
Okay.
So if there's some ideas there, we might use them.
Yeah. All right. So there's the Principia Qualia, which is about consciousness formation and a symmetry theory of valence. So valence is a concept of emotion in the brain where I guess they're looking at consciousness and emotional states. The collective mind and experimental analysis of imitation and self-organization in humans. And then a geometry and functional calculus for Bayesian mechanics, which is more about sort of kind of gets into some Bayesian methods of dynamical systems. So this is about self-organization. So that's...
Is this the same... I kind of missed a little bit of the last thing we're talking about, sorry. But is this all one paper where you're talking about like the symmetry of development of cognition? Or is that just one of the topics?
They're different topics, but I think there are a lot of... It's very heavy on the origins of cognition, it seems like. I was looking at some of these papers. So I'm looking over these. These are the abstracts, and there's stuff on the free energy principle. There's stuff on some consciousness. There's stuff on...
Those are submitted abstracts already?
Yeah, these are submitted. So they put them up here.
Okay.
And then, like, this is what people are going to write papers on later. So it's like several months ago is the last one, but it's still open from what I understand. So. All right, so there we go. So that's all I wanted to talk about with respect to that. So we can talk about that more. I think that'll be a nice thing to talk about coming up. So I see Karan's here. Welcome.
Slack, because I feel like I knew about that or forgot about it, but I really want to submit something to that.
Yeah. Yeah, well, we can talk about it a little bit more and maybe see where we are with that. You know, like, I don't know how long it's open. It should be open probably for a while. It looks like they don't really have enough submissions to have an issue, so they'll probably be accepting for a while yet.
Okay, thank you.
Yeah. Hello, Kumar, or hello, Karan. And Anant Kumar's here. Hello.
Hello. Hi.
Hi.
No. Hi, Bradly.
Hi. You want to give an update on what you've been up to this last week?
Yeah. So far, I've just been, improving the draft of the blog that I'm writing. The description part with some extra images, so I'm clicking images of things that I'm doing. Then changing the like last week, I was trying to, use a different method for feature detection part. Detecting features across the two images so that I can approximate the angle of rotation. So earlier I was using surf, so I was using accurate even though it's tough. So I'm thinking of using some more specific image augmentation techniques to improve the number of features it can detect in for the image itself. Part is also
Karan, are you doing
different techniques.
Karan, are you doing a two-dimensional or three-dimensional rotation?
Yes. The thing is, I'm using two adjacent images at a time. So whenever I consider two images at a time, I give them an approximate, axis about which, they're rotating. And based on that axis, I check the feature and how much it has moved, the displacement of that feature. So let's say, if there was a crevice, a higher pixel intensity or a darker line, on that embryo. So the amount that it has moved, with respect to the approximate axis, using that, I'm guessing the angle of rotation. So the 3D rotation, it's
okay. So it is 3D.
The image itself. Yeah, but it's using data from 2D images.
Oh, okay, got it. I just if you look at the chat, see, I have trouble with pronouncing this name. I'm working. Okay, Jiahang Li and I have a paper submitted on a new algorithm for rotating images. It's only 2 dimensions. It can probably be generalized to 3.
Oh. Okay.
The advantage of it is that it's a reversible algorithm. You may know that if you take a 2-dimensional image and rotate it, and rotate it again and again and again, it gets worse and worse. With this algorithm, it doesn't. Okay. So if you're interested, I can send you a copy of it. It's been submitted.
Yeah. I'd be interested. Yeah.
Okay.
Just I'd be glad to.
Okay.
Yeah. I think it will help in some of the other ones. Yeah.
Okay. Here's my email address. Just send me your email address and I'll send it to you.
Okay. I'll just do it.
Okay.
That's good. So that's what are you planning on doing for next week, Karan? Or
for this coming week, I'll probably have a good projection image, model that I might be able to show. So I'll just improve the projection image generation part so that it can be projected onto the model itself. So I'll be refining the current techniques that I've outlined for that.
Yeah. Okay, that's good. Thank you for the update. And again, as I told Harikrishna, if you need I don't know if you need an extension for the project, but I found out that we can extend them up to 22 weeks if you let me know if there are any problems that you run into that you think you need. Not if you want to add in all sorts of different things, but like if something comes up and you need to delay your submission. So, okay. So we have a new member here, Anant Kumar. Hello. Would you like to introduce yourself?
Hello.
Yeah. Yeah, give a short introduction.
Yeah, I'm a student. I'm student at Amrita right now. So I found this org via Twitter. I stumbled upon it. I got interested, so I joined. Like the Slack and all everything.
Yeah. So what are your specialties? What are you interested in?
So like right now, like I'm working on like ML models and stuff right now.
Okay.
For the time being.
That sounds good. Well, yeah, we have a lot of people doing that sort of thing in the group, so yeah. Well, welcome. You're welcome to attend the meetings and join the Slack and everything. And if you find something interesting, let someone know. And then we can maybe loop you into some sort of project if you want, or you can just So thanks for coming to the meeting.
Yeah, sure.
Jiahang, how are you?
Oh, hi. Yeah.
Hi.
Long time no see.
Yeah. So, do you mind giving an update on GSoC stuff?
Yes. I have some ideas and I have some other updates. So, let me share my screen.
All right.
All right. So, could you see my screen now?
Yes.
Yes. Yeah. I got some updates. Yes, and in last few weeks, I was working with other collaborators, mainly about the data sets, mainly about those microscopic videos, and because it is a little bit difficult to preprocess this video data set and to convert them into the graph structure. So we're still working on them. Yes, it is a little bit difficult. So you will see that the progress is a little bit slow. Yes, we are pushing it.
Yeah.
So besides this process, besides the data sets, I need to make sure you could see my screen. Okay.
Yes.
Okay. Besides those data sets, actually, I see the Wataru and Longhui are working on this microscopy video. So I'm working on some other parts, which is the model design and the motivations of our work on something else. So I will briefly introduce some updates of last few weeks. Yes.
Yeah.
So I make a slide so that you could you could understand my presentation clearly. Yes.
Yeah.
So the topic of the day is the graph neural network and cell developmental analysis, and I'm focusing on the cell tracking.
Okay.
Yeah, and probably you remember that in the last few weeks I've discussed some other ideas, GNN design, which like this. Yes, and today I want to talk about. First of all, I want to talk about the motivations of our work, and then I want to talk about the model design itself, because I found that in the last few weeks I found that this model design is a little bit unreasonable and it can really work well. Yeah. So first of all, we have a lot of questions before the model design, and I think these questions are very important. And first of all, we found that the motivation is still unclear. For example, why do we need to? I mean, the questions here, you can see those questions. Some of them have no, satisfying answers. So I would appreciate if any one of you could discuss these questions with me. Yes, that would be better. And so first of all, it's about our motivation. Why do we need to model data as a graph and employ graph neural network? Although there is only one paper which has thoroughly discussed this question, this motivation, I think we need some more discussions, because although that paper has been accepted by a conference, but I think we still lack sufficient motivation or sufficient answer for these questions, because there's only one paper discussing the GNN and cell tracking or cell developmental analysis. Yes, and I don't have a good answer for this question. Yes, this is the first one. And the second one is about why do we need to consider the topological aspects of data when dealing with cell tracking task. And actually, I think this question is very important, because I think our main points is, first, we want to use the GNN to solve some problems, and the second, we want to consider the topology of data. So we need to ask, why do we need to consider topology? And I have some hypotheses, and the first one is the absolute positions, such as the three-dimensional positions, might vary depending on how observers deal with data, and such as different angles of view or different normalization methods. And I found this phenomenon when I was dealing with the C. elegans position data in the GitHub that provided by the DevoWorm team, and I found that it seems that there are lots of data sources and their positions are totally different. Maybe because their normalization methods are different. And actually, the tracking results of the models should be consistent no matter how absolute positions change, because the embryo is the same, or the target is the same. So this factor will make model learning more difficult, actually. Yes.
Yeah.
This is the first point. Yes. And the second point is why we need to consider the topology, or more specifically, why we need to consider the local topology. Yes, actually, when we consider the topology, we actually do some calculations based on the 3D positions. So it seems like topology can be regarded as a map or function which map positions to some special information or some special outputs. So we can consider the local topology as a measure of the local shape, such as the void or the circle. And my argument is that it is more important to cell. And I would try to use some examples to explain this point. So please wait a moment. Could you see my screen now?
Yes.
Yes.
Okay. Yeah. So my point is, probably you remember that in last few weeks, my argument is that if we have a point cloud, if we have a point cloud, and this point cloud actually are those cells in a certain frame, and given a point cloud, we can explicitly calculate some topological information of this points cloud, such as the persistent diagram and some other kind of tools. This, however, you will find that. However, I remember my argument was consider these points as A, this here as A, and we consider the topology in its neighborhood, such as this neighborhood. And my argument was, given the topology of this neighborhood, we will have some important information of this neighborhood, which will help model to track the trajectories of the cell A. However, this argument is not very clear. I mean, I'm not sure if I explain this clearly, because because the cell tracking task is about the cell developmental process, so we need to consider the time dimension more than just the space dimension. But here we only consider the topology of this points cloud. So I think it's not really sufficient. So I remember I've read some papers which discuss the topology of cell linear tree. Assume that we have a linear tree like this.
Oh,
and
Jiahang, are you drawing on something? Because we see your slides, but we don't see the other thing you're doing.
Oh, sorry.
Oh.
Yes, I'm drawing something.
Oh, okay. So
could you start drawing?
I just see your slides. You might be sharing a tab or something.
Oh, sorry, I'm not sure. Sorry. So you could see you could not see those red drawing.
No, I can't see the drawings now.
Okay.
Oh, there we go. I see it now. Yeah, I see them. Yeah.
Oh, but at this mode I could not draw. I could not doing drawing actually.
Okay, but you see the drawings, yeah.
So just wait a moment. I will first just finish the drawing and I will explain it.
Okay.
Just wait a moment. Okay. Okay. It's I'm not sure if it is clear.
Yeah,
okay.
I guess. I mean, I can see that there are different shapes. Yeah.
Okay. Yes. Yes. My point is that the left plot is the points cloud of a certain frame, and the right plot is a demo cell linear tree. Yes. And this we can call it cross or X. They are actually they represent the cell A and the cell B, and the circle represents its neighborhood along the cell linear tree. Yes. Yeah. So, is it clear?
Yeah, I can see. Yeah, you're mapping sublineages from the cell tree to the space in the point cloud. So it's
yeah. Yes. Yeah. I will make it more clear and yeah.
Yeah.
Just whatever. So actually, my point is that we can we should consider the topology of the cell linear tree more than just the topology of the points cloud of a certain frame. I mean, look at this cell linear tree. Actually, the cell A and the cell B, we can think that they are located in different frames, right?
Mm-hmm.
Yeah, they are located in the different frames. We can think of that. But the cell A and cell B, they might have similar neighborhood in their own point cloud. Is it clear?
Yeah.
I mean, I don't know how to do drawing here. I mean, oh yes, I can do this. Wait a moment. Okay, I mean, let me redraw the whole thing. I mean, just assume that this rectangle represents a certain frame, and we suppose that this frame has a point cloud like that, like this, and this is node A. And first, we consider the neighborhood in the space dimension, which is this, the neighborhood of A. And another neighborhood along the linear tree is this circle, right? Actually, these two neighborhoods, they are totally different, because this neighborhood not only contain the, not only contain the cells in the same frame as the A, but also contain the cells in another frame which is this, which is another rectangle. Is it clear?
Yeah.
So my point is, as for A, there are two different neighborhoods, and the first one is in the space dimension, and another one is along the linear tree dimension, which is a temporal dimension. That means, why do we need to consider the temporal dimension? The topology of the temporal dimension is that because we need to consider the time dimension of the whole things, because we're talking about the cell tracking. That means our final target is to find the absolute position of A in this cell linear tree, right? Although most methods are considering about how to associate cells across different frames, but their final target is to construct such a cell linear tree and to find the position, the absolute position of A, of cell B, and other cells in this cell linear tree, right?
Yeah.
Yes, so we need to consider the temporal dimension. So we need to consider the topology of this cell linear tree. Although probably my explanation are still unclear, but my point is we need to consider the topology of this point cloud in a certain frame, and we also need to consider the topology along this this cell linear tree. That is because we have assume that we have two nodes two cells A and B. They are located in different frames, but probably cell A has a similar neighborhood as cell B, and the neighborhood here is defined in the point cloud instead of the cell linear tree. I call this the space neighborhood, and this space neighborhood. And probably the A and the cell B have similar space neighborhood, and we cannot differentiate A and B based on their space neighborhood because they have similar space neighborhood. But if we consider their temporal dimension, the topology of their temporal dimension, we can differentiate A and B. Yes, I mean, yes, probably, yes.
Yeah, I see what you're trying to do. Yeah, it's basically like connecting the lineage tree, the time information, with the spatial information, and sort of making the connection. So you have this neighborhood of points, and then they evolve in time, and they may move around, and you get like this basically single set, or the single. I guess it would be some topological interpretation of that. for A and for B and so on. So you can track them much more easily. Because that's one thing we've had a problem with is going over time. Like the positions are, we know the positions in discrete points in space. We can average them, but that's not great if you want to look at the time evolution. Because the cells do, you know, they divide, you have daughter cells, those migrate to some extent. They, you know, so the position isn't always clear. Like you can normalize for every time point, but that still leaves you with like this weird weird set of relations. So I think that this is a little bit better way of doing it. You can, I think it's more continuous in terms of the mathematics and in terms of the tracking of the cells.
Okay.
Yeah.
Bradley, can I interject something here?
Yeah.
Motivation. One possibility is to track basilaria and track them long enough so that one sees cell divisions.
Yeah.
Okay. Sorry about that. Anyway, if you track them over, if you track them long enough, you would not only track the individual cells, but you could build up a tree of which cells came from which ones.
Right.
And this could be relevant for diatoms in general, or for any linear, cyanobacteria do this too. For any linear colony, the colony would be, the geometry of the colony would be simple because it's linear. But some colonies develop specialized cells in specialized positions. And so one might be able to work out not only tracking the cells, but building up a tree of when different kinds of cells occur.
Yeah.
Okay, so could be a very important approach. And en route, one could also solve this question of smooth versus jerky movement, let's say of basilaria.
Yeah.
For which there should be lots of data available online.
Yeah. Yeah, definitely.
Okay.
Yeah.
And one other thing, opportunity. Just this morning, we got a proposal for a book on chain diatoms.
Okay.
And they're asking also for a chapter on basilaria.
Oh, okay.
So this could be a contribution, and the time scale would be adequate, I think.
Yeah. Yeah, that would be good. So yeah, this is great work, Jiahang. So this is sort of like trying to solve a problem that you've encountered, and then of course it has applications to other things. So yeah, we have other data beyond C. elegans that we could work on with this too. So this isn't just something we have with C. elegans. This is something we could work on with other things. But for the GSoC project, you know, I'd want to keep it to C. elegans. But I like the sort of conceptual stuff that you've kind of drawn out. Yeah, and then I think like a lot of this, the topological view is very important. I think that's important to keep developing. I think, you know, the need to model data as a graph is largely to see like the connections between them, not only because we have lineage trees, but we also have these spatial relationships. So the spatial relationships, sometimes you have like specific cells and specific locations of a colony or organism. Sometimes you want to have like, you know, there's signaling molecules that are exchanged between cells that are close together or that have like certain functions. So we want to know what those networks might look like. And so we can build networks of this organism, you know, that show these different properties, that show some of these features, like you're showing with the, you know, if we have the topological model, we can show some of those sort of things that you wouldn't necessarily see from experimental data, like, you know, pathways of flow of, you know, physical forces or small molecules or things like that. So there are a lot of ways potentially these could be useful, but you know, we don't worry about that right now. We just kind of like want to build something that will, you know, allow us to get there. And then finally, is it reasonable to consider the cells are in Euclidean space? It could be, you don't. It doesn't necessarily have to be in Euclidean space. You could have some sort of other mapping, and of some other. There are other types of projections that you can use. So we're doing projections with data on the axolotl and the other group. They're taking two-dimensional images and they're projecting out to a three-dimensional image. I know the embryo is originally 3 dimensions, but you're making sort of a projection outward. And so to do that, they're doing some things with topology as well, just not calling it that. And so, yeah, I think that you can use other types of space, like there's Euclidean space, other types of like projections that you can use that are not Euclidean, that are curved. And so that's something also that can be used, although it's a little harder to work with for like, looking at, you can look at different, you can look at the connectivity in Euclidean space. You can get certain information about like a shortest path, but you can also get other types of information. You can look at curvature and things like that, which occur in the embryo. So, that's useful as well.
Yeah. I think the topology is our first question, and if we consider, is it reasonable to make the series in the Euclidean space? I mean, I remember there are some papers talking about the series in, the embryo and some other kind of space, such as the manifold or such as the hyperbolic space. But I think it is another question, and maybe we can discuss it later. But I think it is not very important by now.
Yeah.
Yes. So I think as for now, I think for now I will focus on, first of all, the GSoC projects. We need to make sure that we will have an effective graph neural network model which can be employed on the C. elegans data, and to have some reasonable outputs. And my second focus is that I will try to combine the topological data analysis with graph neural network, and to see if we have any new findings. Yes, but yes, I will focus on these 2 points for now. But actually, I have 2 questions here. First, I think I'm not sure are these questions. Oh, sorry, I'm not sure are these questions important. Wait a moment. The first one is the topology of symmetry of a certain organism a variable.
Yeah, invariant. Yeah.
Yes, is it true or not?
Is the topology invariable? I don't know. Well, it's a hard question to ask, because we're just looking at like an average of many organisms in the data that we get for cell tracking, or sometimes we're looking at just one. And so knowing like what the variation is across the different individuals in that species, or that's a problem that is probably going to make it not invariable. So like if you want to find like a sort of a platonic tree that like fits every data. They do this with different models where they like sometimes they'll do it with the human body. They'll have like a model of like how the shape of the body, the specific gravity of the different parts of the body, like different tissues. They'll use like some they'll do some averaging, and they'll kind of come up with an average, but it's not perfect. You still have these you still have a lot of variation around that average. And you see this with like embryos. You have in C. elegans you have specific mutants. You have different in C. elegans we work with it because it is more or less invariable as embryos go for different organisms. But like you still have a lot of variation in C. elegans. You have a lot of variation between males and hermaphrodites, between different mutants, between other just environmental factors that maybe give a introduce a tiny bit of variation, but it's not something. My point being is that I wouldn't worry about like if it's the perfect model. It's just kind of like, I think actually a better approach is just think that it's like a generative thing where if you generate a model, it's like one possible solution, and then like maybe there's like some you can generate different models that are similar, and that's like basically what you're dealing with for that species. That you generate different variations on it. It's not too much different, but that's what it looks like. So then the second question is the topology of a cell tree of a model organism analogous to most other organisms? So that's another thing. Again, when we apply this to different organisms, sometimes it's vastly different. Like the Basilaria, which is an algae, it's much different than C. elegans. So it's going to have a much different topology. It's a colony. But the point is that you should have like, if it's multicellular, maybe it should have certain properties. But that's as far as the similarities might go. If we look at a species that's closely related to C. elegans, it might have very similar features in relation to some of the anatomical parts. But again, the anatomical parts might be related. There are commonalities in development across different embryos, depending on where you are in the tree of life. But those commonalities, we should be able to pick those up. And that's kind of the point. It isn't about like replicating other organisms. It's about finding things that are in association. So if there are certain parts of the anatomy, like, muscle groups or, a connectome, those should be, you should be able to identify them with the method. So it's analogous in the sense that it'll pick up structure, but, beyond that, it's hard to really make direct comparisons across to other species.
Yeah, I know, I understand. Yeah, I asked these two questions because the first one is that I was discussing that the topology of a cell linear tree, but because the model will learn from a lot of data, and if the topology of a cell linear tree is fixed, that means when the model learns from one cell linear tree, then the knowledge from this cell linear tree will be employed on other data, other microscopic video of this organism, and they will help this model to track cells, even though this model doesn't have the cell linear tree of that microscopic video. And the second question is that probably their cell linear tree will not be the totally same, but I'm not sure. If it is transferable. It's like the idea of transfer learning, because actually, as for a lot of data sets, what we have only some cell trajectories instead of the whole cell linear tree, such as those data sets in the cell tracking challenge. So if we do not have the whole cell linear tree, that means we cannot obtain the topology of this cell linear tree. That means so I would wonder if we have the cell linear tree of a small model organism such as the C. elegans, if we can transfer knowledge of this cell linear tree to other more complex organisms which have no cell linear tree. Yes. So yes, that is why I asked these two questions. The second question is like what we do on the transfer learning, which is to transfer knowledge of an organism to another organism, and these two organisms have some difference, but they also have some similarity. Yes, that's why we do the transfer learning. Yes. Yeah.
Well, I think that's a good question. Yeah. It's I think that I mean, there are going to be some things you're going to be able to transfer and some things you won't. So like any kind of cell division, we're going to be able to, if it's like one mother cell to two daughter cells, we should be able to do some, it should be able to, considering, if you're like trying to extract it from input data, if the mode of division is basically similar, then, it should be able to transfer some knowledge to that. If it's a different, if the mode of division is different in some ways, like, in the image, like visually, I guess you'd put it, then, it's going to learn less. But it should have there should be some applicability. So you should always be able to transfer some of what we've learned in C. elegans to other species if we apply this method. But it depends on a lot of things. There's a lot of variation in development. So I think it'll work with other organisms. I'm not really sure which ones it'll struggle with. We could probably make guesses as to that. But I think there'll be some transfer learning. It's going to really depend on like the specific species. But then, if we want to use it on a specific species, we can, retrain it, give it more information, etc. So that's something that'll just be like something else have to be overcome as we go. Yeah.
Okay, Bradley, can I make some comments here?
Yeah.
3 of the points. Topology of a lineage tree, is it invariable? There was a paper, I think in the 50s or 60s, on cloning frogs. And they found the assumption is that cloned frogs are all genetically identical. Okay, and what they found is that the pigment patterns on those frogs were different from one individual to another. Okay, and this kind of work has led to there's a small literature on variability in development, stochastic processes in development.
Yeah.
Okay, so there's information about it. I don't know if that applies to nematodes. Okay. Second, applying the cell lineage tree to other organisms. Well, maybe not. But I'll give you an interesting example. There's a cyanobacterium that develops. Cyanobacteria generally consist of 2 cell types, vegetative and heterosis. And there's a colonial cyanobacteria that forms a line of vegetative cells with heterosis at the end. And as it grows, it forms a pair of heterosis in the middle. And then it splits between them into 2 colonies. Okay, so that kind of data is probably available. It's an example which might be fun to follow as a cell lineage tree. And the last question on the Euclidean space, I was trying to think of that. And if you take a multicellular organism that has differentiation waves in it, you could use the time at which a wave impinges on a cell as a coordinate, and that might be a non-Euclidean space. I don't know.
Okay.
It would be when does the cell participate in a particular wave? Might be a new coordinate. No, I'm having trouble thinking outside of Euclidean space, so that would be time plus its position. So it's that's 4 coordinates.
Yeah.
Euclidean. Okay. But it's an example of possible consideration of the non-Euclidean space by considering time variable.
Okay.
In a way, the development of an embryo is similar to relativity. You have 3 space coordinates and 1 time coordinate.
Yeah.
Even though it's much slower, nothing near the speed of light. Okay.
Yeah.
Okay. Oh, by the way, you've mentioned a paper in the 50 or 60s, right?
Yeah, I don't know if I can try. Yeah, send me your email address and I'll try to dig it out. I might still have a record of it. Okay.
Yeah.
But it was fascinating because it showed that development was variable, even if the genetics is not. And I think the laboratory conditions were as constant as they could keep them at that time. But okay.
Okay.
Yeah.
I see. Okay.
So, Jiahang, one final question on this. So, how are you planning to move forward on some of this? Like, this is, like, you know, I mean, are you having any trouble, any obstacles? What is the?
Oh, you mean the plan of the GSoC project?
Well, I mean, you know, like, what are your next steps? Like, I know you wanted to clarify some things of the model, but what are the next steps that you want to do for this part?
I'm sorry, I didn't really get this.
Oh,
what was that?
Well, what are the next steps for this? Like, what are you planning to do in the next few weeks?
Okay, next few weeks. Actually, because it is August now, and I'm not sure this idea will really work, but we need to make the GSoC project. So, actually, because there is few works regarding employing GNN graph neural network on the cell developmental process. So my plan is to we have only one implementation, which is to employ GNN to track cells, and that was the paper that I've mentioned, which was accepted by the EWCV conference. And I'm not sure is it okay to re-implement that implementation as such as the API or something else as the implementation of our GSoC project.
Well, yeah, you could re-implement it. I think it might be, you know, nice if you could make some modifications to it to reflect some of the things we've been talking about. But I don't know how hard it would be to do that. I think, yeah, but you can use that as a guide. I would definitely not want to reinvent the wheel if we don't have to. So, being able to just have a nice framework, and so this is an open source platform or this is the work that they have is open source, correct? Or
Mm-hmm.
Okay, so we can,
Yes.
yeah, I mean, we can modify it and make modifications to it and see how that goes, and see what the performance is like. And then,
Okay. Yes. Yeah. Okay. Yeah.
because a lot of
Yeah.
GSoC.
I will make.
Oh, go ahead. Go ahead.
Okay. I will make sure that we will modify the original implementation and make it like a platform or framework, and people will find that they would easily use this framework. Yes.
Yeah.
Yes.
Yeah.
I will do some benchmarking on some data sets. Yes.
Right. And then, we can actually add in things like the topological aspect. I mean, there's a lot there. It's just kind of like a huge huge chunk to take on, which can be done after the project. But, we have to have a nice baseline to start from so we can add on to it. And I think that, if you're using something that's already kind of been developed and it actually works, and then you can modify it, that would be a good way to go, because you'll have at least some core, functions and things like that can be used.
Yes. I know. Yes, I understand. But actually, the most difficult part by now is not the model or the GNN part. It's about the computer vision part. It's about how to pre-process those microscopic video. It's more difficult than what DevoWorm has done. Yes, it's not just simply employ the ResNets or some other CNN on the microscopy video. Yes, we need to make sure the quality of the outputs of this CNN is good, is satisfying. Yes,
Yeah.
I mean, we're working on this. It is very, actually, I think it's very difficult. Yeah.
Yeah. Like I said before, the first part is gonna be the most challenging part, because you want to be able to have good input data for the next part. If you don't have good input data, that's
Yes.
a problem. That's the
Yeah. Yes. Yeah. Actually the implementation of constructing a graph data sets based on the given the given point cloud or something else has been has been done. Yes, I think it has been done in the June or the July. I don't remember. It's not very difficult. Actually, the most difficult part is how to process those video data. Yes, and I remember the Wataru and Longhui, they are working on this part, and they are still diving into the details of the implementation of that paper, of that GNN for the checking paper, because they've used a lot of tools and computer vision tools to implement such parts. And I found their implementation are very hard to describe it. They are very difficult. Yes, they are very hard.
Yeah.
Yeah. Yeah. So we'll make this process faster. We will push it. Yeah.
Okay. Well, that sounds
Yeah.
good. Yeah. Thanks for the update and discussion. That was good.
Mm-hmm.
Okay. I don't know if we had anything else we wanted to talk about today, but I think we're at time for this meeting. It's like Jesse had to leave, and Harikrishna had to leave, and we have some things in the chat. Let me go over those real quick. Okay, it's just a matter of like people saying goodbye. Okay. Yeah. So thanks for attending the meeting. And I'll send out some information on some of the things we've talked about, those abstracts, so that Dick can read through them. And if you have any questions, you can catch up on email or Slack. Yeah. Okay. Thanks.